University of Washington's eScience Institute. As this is part of the University of Washington, I want to begin by acknowledging the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Squamish, Tulalip, and Muckleshoot nations. As we begin, I'd like to remind our in-person folks to turn off their cell phone. I love getting to say that again now. We have a microphone at the front of the room for the Q&A period. If I could ask the speakers to repeat the questions so that folks on Zoom can hear it. And if you're joining us on Zoom, please use the Q&A feature via the bottom on the button of your viewing window. Our speakers today are Dr. Tucker Bergen and Orion Dollar. We'll start with Orion Dollar. Orion is a PhD candidate in chemical engineering at the University of Washington. His work involves employing artificial intelligence to enhance the speed and efficiency of functional materials discovery. He has a particular focus on multimodal methods, including merging things from molecular structures and properties and databases, scientific literature mining, and putting this all together in a unified model framework. He holds a master's in environmental engineering from the University of Kansas, and his talk today is attention-based generative models for de novo molecular design.
Cool. Thank you, Dave. Yeah, I'm a third-year student at UW under the Fentner Research Group, newly minted PhD candidate, finally. And I'm going to be talking mostly on my work on the generative models for de novo molecular design, specifically for functional materials discovery. So there's a lot of urgency behind materials discovery, right? There's a lot of different problems that we're being faced with, disease or rising drug prices, sort of endemic viruses that we're all, of course, very familiar with, rising temperatures. This lady here is clearly very sweaty due to climate change. So all of these problems sort of have a common theme, which is that they have a materials-based solution. So if we can discover the right material, we have a chance at sort of addressing these problems from the ground up. And there's a couple different ways that we can go about designing a molecule for a given application. And it involves mapping a chemical space to a functional space. Sort of the standard way to do that in the past is known as direct design. So this is where we make a small modification to an existing chemical motif, and then we perform some analysis on it. That could be an experiment or a simulation, and we make a direct measurement and make a modification to the motif that we're testing based on that measurement. However, direct design is typically a very slow process. So to speed it up, what we'd like to be able to do is map that in the inverse direction, where we actually define our target in functional space and map that back to a molecular structure. So in this way, we can sort of screen or test hundreds of thousands of molecular structures at once and generate those that are most likely to exhibit a given functional property. I mean, there are a couple of different ways to do that. One is just sort of brute force, high throughput virtual screening. But with the advent of deep learning and AI, there's actually some basically direct optimization methods that we can use to sort of find the correct molecular structure. So if we look at the full life cycle of materials discovery using AI, there's a lot of different components that go into it, right? So we need to create a curated data set. We need to design some sort of molecular representation or design some molecular feature. We might need to do some property prediction. But what I really want to key in on today is the generative method. So if we're able to basically use an AI to develop novel molecular structures and optimize within the framework of that AI itself. And there are a lot of different ways to go about this as well. But again, the one I really want to key in on today is the variational autoencoder, which essentially maps a probability distribution of molecular structures to a numerical probability manifold. And we can use this manifold to optimize the structures within the model itself. So just to sort of go over the VAE architecture in a little bit more detail, it's essentially a variation on the standard autoencoder. So the model is mapping an input to an output, and our inputs are molecular structures. And as it does this, because there's this bottleneck in the center of the model, we learn a numerical representation of our molecular structure. And we can actually use that numerical representation to decode back into the original structure. And the loss of the model is how accurately the decoder mapped the output back to the input. So we're just simply recreating the inputs to the model. And you might be thinking, this is sort of like a trivial task, but if we add this sort of variational component, sort of the value add of this model is this really rich, meaningful latent space within the center of the model. So to do this, we add this KL divergence term, which is essentially mapping the numerical embedding of our molecular structures to a Gaussian distribution. And when this happens, we get a nice sort of Gaussian distribution around each of our data points, which we can interpolate between and actually decode points the model has never seen before back into a valid molecular structure. The scale divergence is going to be, stochastic mapping to a Gaussian distribution is going to be important in some of the stuff I show later. So I just sort of wanted to introduce that concept a little bit early. When we're actually working with molecular structures, we need some way to design a feature representation, right? So if we take a molecular structure, what we can actually do is turn it into a string-based representation that we can feed into our model and use sort of a standard natural language processing architecture to use in our model. So what we can do is just sort of work our way through the molecule and design different tokens that represent different sort of branching patterns. For instance, if we branch, we can use parentheses. We can have different tokens for aromaticity or different atom types. So here, a lowercase carbon would represent a aromatic carbon in the structure, or the 1 would represent a ring opening and closure, and so on and so forth. And we can encode our entire molecular structure as a string. And during training, what happens is we get our sort of unique numerical embedding as we pass our molecular structure into the encoder. We pass that back through the decoder and we can reconstruct the molecule. And as we train, we get a unique numerical embedding of each of the structures that we pass through the model. So this is sort of the value add that what we learned during training. The other thing we can do is we can sample an entirely random numerical embedding, and we can pass that directly into the decoder. So here we take a numerical embedding which has no associated structure prior to passing it into the model. But as we pass it through the decoder, we get a brand new novel molecular structure that was not previously in our database. So in this way, we have a generative model. Of course, we can't just sort of out of the box use these models right now. There's a number of problems associated with them. One being posterior collapse. So this is when instability in training leads to the decoder essentially ignoring the numerical embedding. And in this case, we sort of get the same molecular structure representation out of the model at all times. Another thing that can happen is syntactic errors in our decoded SMILES strings, leading to actually invalid molecules, meaning that we're missing a ring opening or closure. We have a wrong valency, in which case the molecule is no longer valid. And finally, we have sort of a general lack of interpretability about what the model is learning. And we have sort of few well-defined properties for downstream optimization tasks. So both of those problems are not trivial, and sort of why you aren't seeing these models used sort of standard in experimental labs to sort of expedite their design process. So we asked basically a pretty simple question, is that because this entire model is sort of formulated as a natural language processing task, can we improve the molecular design performance by updating our architecture to reflect the state of the art in natural language processing? So for those of you who are familiar, attention is sort of a very popular sort of novel mechanism that was established in the last couple of years. So we basically took an attention mechanism and we added that to sort of the standard VAE architecture in a couple different ways. One with this RNN attention model and one with a sort of purely attention-based transformer model. And we trained these on a molecular data set to see how their performance sort of compared to one another. So just to go over real quick some of our training details, we had a number of different dimensionalities for our model. We tested on 2 different data sets. Zinc is a sort of small drug-like molecule data set, whereas PubChem has somewhat more complex organic structures. And we evaluated on a number of different evaluation metrics. And I kind of want to split them into 2 different categories. So there's reconstruction accuracy, percent validity, and cross diversity or model exploration. And these are all sort of sampling or sort of downstream model metrics, which is how well is our model creating new molecules. And then the last evaluation metric, the latent information density, is essentially a metric of the internals of the model. So how much information is the model storing in its latent bottleneck from the sort of training distribution. So just to sort of summarize some of the results, if we look at the zinc data set, the molecules are typically a lot smaller. So we have sort of an easier task for the model to learn. Whereas with PubChem, we have a much wider distribution of SMILES lengths. And this is sort of representative of a really good task that attention can handle, because attention is basically no longer restricted to local environments within the molecular structure, but it can learn these long-range dependencies. And when we evaluate the reconstruction performance, we actually see that the attention models do perform much better on these larger PubChem molecules. And we can see a pretty significant dip at around token sequence position of 80, where our data is sort of becoming very sparse in our training distribution, but the attention models still perform very well on reconstructing these molecules. So what we can sort of take away from this is that the attention VAEs are better at reconstructing large molecules, and by proxy, we sort of can infer a higher quality latent space near the tail of our distribution, or sort of near the outskirt of our training distribution in general. Meaning these models are better at generalizing to sort of small bits of data that occur in the training distribution. The other thing that's really nice is that we can sort of try and interpret these attention weights or attention heads that exist within the model. For the transformer, we see that there are this very nice, unique syntactical grammar that's learned within each of the heads, and that these patterns actually sort of correspond to valid molecular fragments or substructures. So we can see here we get an aromatic cycle extracted from our attention head. We get this aliphatic carbon cycle, and we get some sort of branching pattern. So we can see that the model is learning sort of valid molecular substructures, whereas there's actually another function within the RNN attention that the attention head is learning, which is sort of the most efficient compression. So depending on the way that we design our architecture, the attention heads might perform sort of different functions within our model. So to sort of summarize where we've gotten so far, our models with attention have better or more accurate latent embeddings. So we have a better numerical representation of our molecular structures. The transformers show the potential to have these interpretable molecular substructures that we can extract, potentially, learn chemistry from or use in downstream modeling tasks. And our RNN with attention has this very efficient compression of information within the attention head. So we want to ask ourselves, how does this reflect itself in the sampling behavior of the models? And there are two things that we really care about, right? We want a very high validity of molecular structure. So every time we sample, we want to make sure that it's a valid molecular structure. And we also want a large diversity. So we don't want to just simply recreate the training distribution over and over again, but we want to create new molecules that the model has not necessarily seen before. And as it turns out, both of these are a function of the information density of the latent space, which I mentioned earlier. So sort of as a high level overview of what this means, we can actually look at the encodings that the model is creating for a subset of our data. On the x-axis, we have each latent dimension. So we have 128 latent dimensions that are storing numerical information about our structures in the center of the model. On the y-axis, we have 50 unique molecular structures. So we see sort of a structural footprint of the latent bottleneck within our model. And remember, because we have this sort of stochastic Gaussian distribution that we're enforcing, there's actually a couple different ways that this information can be stored. We can store it as a wide distribution of means around a small standard deviation, or a tight distribution of means around a large standard deviation. Well, what does that mean? It essentially means that we want our data to look standard normal, and it can encode that in the mean vector, or it can encode that in the standard deviation vector. If it encodes that in the mean vector, what that essentially means is that we have a lot of high quality data within that dimension. And we can actually see, if we take this for our 50 samples, that we start to get this standard normal representation directly from the means, whereas our standard deviations are very, very low. So when we sample and we enforce that standard normal within our dimension, that's all being stored within the latent mean vector of the model. Whereas on the other hand, in this other dimension, almost all of the means are 0, but we have a very high standard deviation. So when we sample from a Gaussian distribution, we're still getting a standard normal, but none of that information is being stored within the latent means. And we can actually measure this with Shannon information entropy. So what happens if we probe these sort of different kinds of dimensions? Well, if we sample directly from the low entropy, or basically the dimensions where there's this orange dimension where there's no information being stored in the latent mean vector, we see that there's no information, right? So as we sample from this, the original structure is unchanged. So the model is essentially ignoring this dimension and sort of trivially assigning it to the standard normal. Whereas if we do the opposite, we only sample from the high entropy dimensions, what we see is this nice, diverse array of molecular structures, which is what you want to see. So we can see that the model is sort of picking and choosing which dimensions it feels is meaningful, or it wants to store information in, and sort of ignoring the rest. And yeah, all of the meaningful structural information is stored in these high entropy dimensions. So we can actually create sort of a unified metric or a sum metric where we can sum the Shannon information entropy across every single dimension, and we can use that to analyze the sampling validity and diversity of the model. And this is the total information density of the model. So if we look at the information density as a function of the training epochs, we can see that the transformer models at epoch 100 have the highest information entropy, or again, the most complex representation stored within the bottleneck, whereas the RNN with attention has the lowest, meaning it has the most efficient compression of information, sort of like we were talking about when we were analyzing these heads directly. And if we look at the latent memory entropy as a function of sampling validity and model exploration, we can see that there's some very clear trends that arise. So the more information that's stored in the model, the harder it is to sample from, right? We're sampling from more dimensions, and we're sampling more and more invalid structures. Whereas on the other hand, as we increase the latent memory entropy, we have a higher cross diversity, or a higher amount of exploration. So because we have all this extra information stored, we can sample a wider diversity of molecular structures. So this parameter of the internals of the model is actually sort of directly optimizable based on these different sampling criteria. And what's nice is that since this work has been published, we have novel input representations that actually guarantee 100% validity in the form of SELFIES. So we can sort of take this information entropy and use it as sort of a direct proxy for optimizing the amount of exploration that occurs within our models. So in conclusion, by adding attention to these models, we've gotten a high reconstruction accuracy, meaning a high quality molecular embedding. We have an interpretable sort of attention pattern that occurs within both the RNN attention and transformer models. One in which we have the highest efficient compression of data, with a lower dimensional latent space, whereas the other is extracting these interpretable molecular substructures with the most information dense latent space. And in terms of future work, we plan on applying this to a couple of real world use case studies, peptoid sidechain discovery and organic corrosion inhibition, as well as sort of tying this back into the sort of entire life cycle of discovery by mapping these numerical embeddings of molecular structures to other numerical embeddings that you can gather through sort of other machine learning pipelines, such as natural language processing. So with that, I'd like to thank my group, my PIs, David Beck and Jim Fentner, and open it up for any questions. Evan. Yeah. In terms of the sort of practicality of these models as they stand, like RNN attention is probably better. I think in terms of like algorithm development, and if you want to play with sort of interpretability, then transformer is better. One thing we're looking at is like how these attention heads or these attention weights change when you start predicting a property. So can we learn something about molecular structure relating to, like log P or electrostatics or something? Can we learn from the attention weights in the model itself simply by predicting that property? So that's something you could only do with sort of the more interpretable version of the model. But in terms of just like sampling and generating molecular structures, the RNN attention is probably better. Yep. Yeah, I mean, so that's definitely an active area of research. It's I haven't found any reason to think that the Gaussian is insufficient in modeling that latent space. But I know like there's definitely people have used other sort of distributions or sort of kernels within their latent space. I can't think of what those models are called off the top of my head, but in terms of the VAE and sort of the development of that architecture, the Gaussian has been sort of the standard since like 2014-ish. Yeah, so it actually definitely does. I can actually show you. What's that? Oh yeah, sorry. Tucker asked if changing the input representation from smiles to selfies might change the exploration of the model. And we can see that it definitely, actually, I mean, it 100% does. We've tested it. The selfies in general have a higher, more complex sort of latent space representation. And that same pattern persists when you change input representation, meaning the cross diversity is higher too. So I'm not sure exactly what it is about the selfie that prompts more exploration, maybe more advanced relationships within the representation itself that the model is capable of picking up on. But yeah, it definitely has effect. From what I've seen, yeah, selfies are superior.